Howdy again. This is Jim G from Texas A&M University. In today's talk, we will have an overview of some of the basic concept in medical image processing. Imaging start with data acquisition. Then through the reconstruction, we turn the signal into images. Then oftentimes with those images, we like to manipulate the images and extract the information or make it much more easier to interpret. And that is due through this process called image processing. So there are many reasons we might want to process a medical image. Sometimes we like to kind of reduce the noise or enhance the contrast. These are typically called the low-level image processing. Sometimes we like to extract the interesting features. For example, separate the tumors or blood vessels or cells or bones, so we can measure some of the features from here. In some other cases, we like to correlate images so we can fuse the information. This is called image restoration or image alignment. Then in other times, we would like to extract the quantitative information from the images. This could be, for example, to measure the size of the brain, the velocity of the blood flow, or concentrations of drugs. These are called quantitative analysis. So in this lecture, I'll go through this individual applications with some brief examples. So lower level processing, here's the example. This is a figure that's been used often in the image processing field. We could have images that you can do a low pass filtering, and typically when you do a low pass filtering, you lose some of the resolution, but the image will look smoother and less noisy. You can apply a high pass filter. The high pass filter will make the edges more outstanding, but it also kind of enhances the noise. And there are many different ways to do denoising. In addition to the low pass filtering, we have a lot of adaptive filtering or edge preserving filtering, wavelet based processing that can oftentimes performs better. These are the topics that will be covered in the digital image processing course, so I will not dwell too much on that. Those filterings are typically done by multi-dimensional convolution. In this example, I'm showing you doing a low pass filter. As you can see here, you convolve the image with a two-dimensional filter. It's almost like averaging, and so from the left, after the low pass filtering with the convolution, you get that image output. So this can be turned into a frequency domain operation, because convolution in the image domain will be turned into multiplication in the frequency domain. So you can multiply with the Fourier transform of the filter in the frequency domain. Oftentimes we like to manipulate the contrast of a figure because maybe it's too dark, or that we like to adjust the contrast or color. This can be done typically by looking at the histogram. Histogram basically is a statistical tool for you to count the number of pixels with a certain intensity. Here, what you see here on the horizontal, in this histogram, the horizontal represent the intensity, and the height of the vertical bars represent the number of pixels in this intensity. So if you have a picture like on the top left one, relatively dark picture, you expect to see a lot of high counts in the lower intensity range, just like what you see here. You can do a nonlinear mapping of the intensities through a function just like what you see on the top right. On the horizontal, this is the input intensity. On the vertical, you have the output intensity. As you can see here, you basically lift up the intensity, lower intensity level very much. And after this histogram equalization, the histogram for will be kind of equalized, meaning approximately have an equal number of pixels in different intensities. As you can see here, the equalization indeed makes the image easier to see. This is another example of histogram-like equalization, that you have an image on the left that is very dark, difficult to see, and with the equalization, you can see in the middle, you could selectively doing equalization like what you did on the right. The contrast enhancement, sometimes you can do the other way. What you see on the left, this is a digital mammogram. You can invert the intensity so that sometimes it's easier to see this calcification. We can, of course, zoom in or zoom out the figures, images, so you can see it more easily, the small features. Many other techniques that being embedded most time on the medical scanners. They usually being provided as processing tools or on the PACS system for the doctor to manipulate the images. In the research lab, oftentimes we can use the MATLAB image processing tools or the public available software tools like ImageJ to do the common processings. So I'm gonna move on to different specific topics. We'll start with this image segmentation. Segmentation is to partition the image into subregions that as shown in this burn image segmentation example. I'm doing a very simple segmentation here. I just separate the white matter in the middle and the gray matter on the right. Those subregions usually are not do not overlap with each other, and there are certain common features that make the region homogeneous, so you can differentiate one region from the other one. Okay, there are different ways you can do segmentation. Sometimes we just interested in extract the boundaries of a region. In this case, we're getting the boundaries of the cardiac at the heart wall, segmentation has many applications. Quantification actually is one, because with the segmentation you'll be able to measure the size, you'll be able to measure the shape of the certain tissues or organ that has a lot of diagnosis value. For example, you can measure the brain atrophy of the Alzheimer's disease patient, or you can measure the size of the tumor, so you will see whether it's growing or whether it's shrinking. So you can have a quantitative assessment of whether treatment is effective. With the segmentation, we can also do visualization. For example, you can segment the bones of the CT image, then you'll be able to reconstruct the 3D bone structure You can do the same thing for the brain or the vasculature. With the segmentation, you could do many other things that I will not get in detail. But here is another example with the blood image. You can segment those, and then you'll be able to count the number of blood cells. This example. With respect to the edge detection, there are many different ways that you can do. Again, these will be covered in the digital image processing course, but there are algorithms that be able to be more robust to the noise than the others. The challenge of the image segmentation in the biological or medical image processing is that oftentimes the biological system contains very complex structures. Unlike the natural photograph that you try to segment an animal or a car or a building, in the biological tissues, the shape of the organ that you try to segment often are very complex. Also, the signal-to-noise ratio often are limited, and the problem therefore sometimes is ill-posed. Now, what does that mean? Here's the example. So this is a black-white photo. This is not a medical image, but it's just black-white photo. If I ask you to say, hey, segment this image for me, it would be very difficult because the features not salient and it looks like very noisy. But however, if I show you this figure or image and then get back to this figure, I say, hey, can you segment this? Use your hand. It becomes a much easier job because now you have a prior knowledge of where that dog is. But without that prior knowledge, this problem is ill-posed because there may be many different solutions. And those prior knowledge can be applied to assist the segmentation. One example of using this prior knowledge is the template matching. Template basically is the standard image or structure with a known segmentation. We can match the template to an image, therefore you would have automatic segmentation. The template could be a volume template like a standard brain. It could also be a shape, like a shape of a heart or some other structures. A very well-known algorithm to apply those template kind of matching is active contour. In the late 80s, this algorithm called SNAKE. It's a contour that can change its shape to match with the shape in the images. Recently, this been evolved into a method called level sets. It's a much more powerful, capable algorithm that can handle very complicated shape, overcome many of the problem with existing earlier SNAKE method. So if you're interested in the shape or edge surface detection in the medical image analysis, you should look into the level sets. Well, we really don't have time to get into the details, but those active contours are one-dimensional features that are governed by the differential equations. It has a elasticity, internal force to make it change shape. You have also external force that will be defined on the feature of the images. And if this works well, it will eventually converge to the edges. All right, and then the next topic we can talk about is image matching or image alignment. The technical word for this is image registration. Imagine that you have two images looks like there's two images that you can see here they're actually coming from the same subject. They're just in a different orientation, different locations. This might be two images contains complementary information. So when you align them, you would be able to fuse or integrate the information from the two images. So why this is important, you may ask. Let me give you this example. So here what you see is image of the abdomen. On the top, this is the CT image. Okay, you can see the bones, some of the anatomical structure in the body. In the middle here, this is the nuclear medicine image, a PET image that highlight the cancer. Okay, but without anatomical reference, it's very hard to tell where that cancer is. By fuse these two images together, or register these two images together, you can create this image on the bottom where that you have the anatomical reference and also the highlight of the tumor. So this clearly application that you can do with image fusion. This is being used on the PET-CT all the time. In some other examples, you're doing a dynamic study, so you acquire images at different times. This is the contrast enhanced animal cancer study that you would what you see here. There's a different intensity is due to the contrast agent uptake. So in order to analyze those drug kinetics, you need to be able to align these images. So you would compare apple with apple. In this last application, sometimes people do population-based study. You study the variation of the shapes of the brain, for example. You need to align them onto the same coordinate reference. That's called a morphometrics. How do we actually do this? Here's another example. The functional MRI we talk about. In the functional MRI, you typically take the images while the subject is doing nothing. This is called a baseline scan. And then you will acquire images that the subject is doing some functions like a reading or speak or motion. And then you compare the baseline with the active images, and you will see which area has a significant change that will be correlated with the actions you're doing. In this example, this is a finger tapping application. Of course, it's critical in this kind of application that there will be no motion between the stage 1 and 2 or the baseline, the active. So you will have you will be able to compare the changes accurately. In the old days, image alignment can be done, and that was still being done in some cases with stereotactic markers. You basically have to drill a hole and screw some of the markers on the patient, and these markers will be highlighted on the images that are being acquired, and then by comparing those marker locations, you'll be able to derive a coordinate transform that will bring the images into alignment. We're actually more interested in this course on this so-called retrospective image registration. So we acquire the images without these markers, and we try to bring the images back into alignment. So how do we do this? So when we do this alignment, what we need is the first thing is a spatial transformation, the rotation, the motion, the moving along in different directions, sometimes stretching along different directions, and then we can look at the two images after one is being rotated and moved, and look at how similar they are. Okay, this needs to be a mathematical similarity measurement of the two images, and then based on the similarity, we will try to get back and to move the different to a different location, do a different spatial transformation, and then compare again. This is like you can imagine that you have two images like on a film, that you can look at them, overlap them, and look at them and try to manipulate them and bring them back into alignment. All this being done, of course, on the computer, as long as you have these three critical components: the spatial transform that is powerful enough to accommodate the differences between the two. Then we have the similarity metric that can measure the structure similarities between the two images, and then you have optimization method that can search for the optima. There are hundreds of papers on this topic. Again, on the image processing course, we'll cover some of this material. There's a graduate level course called Advanced Signal Processing for Medical Imaging, ECEN 617, also cover some of these topics. Finally, let's talk about the quantitative analysis. In the medical image analysis in particular, oftentimes we like to have accurate numbers, so you will make a more precise diagnosis. This many times involves a measure of certain physiological or pathological quantities. For example, the blood volume in the heart can, during the contraction relaxation, can tell you the function of the heart. The functional activity in the brain will tell you whether the neural system is working correctly, and the concentration distribution of the drug dose will tell you whether the drug is being delivered to the correct location. Those quantitative analysis usually involve a model. For example, the function fMRI assume brain activation is associated with the blood oxygen level change, and then we build a mathematical model to account for the blood oxygen change and the blood flow, relate those with the images that we acquire with the MRI. Then inverse solve that inverse problem. You find out the functional activation. Oftentimes this also involves statistical signal processing, the student t-testing, the model fitting, sparsity-driven regression mathematical tools. So without getting into detail, just let me give you a couple examples. This is some of the early research being done in my group. In this first example, you're looking at the breast dynamic contrast breast MRI. So you acquire a sequence of images while the contrast agent is being injected. You look at how the contrast agent being perfused into the tissue, and by having a mathematical model account for the blood flow, and how fast the contrast agent be injected, and how that contrast agent will be exchanged between the blood pool and the intercellular space. You'll be able to solve that and eventually generate a parametric map. Tells you it's called a K-tron, tells you how fast that exchange happens between the plasma and the intercellular space, and those parametric map can be used in further to infer the cancer, make the cancer diagnosis. In this second example, what you're looking at here is Doppler ultrasound. Doppler ultrasound images. This is an image of the carotid artery on the neck. This is the main artery that supplies the blood to the brain. Because the motion of the blood cell create a frequency shift or Doppler shift that are proportional to the blood flow velocity, and that will be reflected on the demodulated RF signal, and you can actually measure the velocity. With the MRI we can do the similar measurement, just a different kind of physics, but you can measure those blood velocity and the motion. So in summary, the medical image processing can make it easier to perceive information from acquired images, or to facilitate image analysis to extract features or quantitative numbered information for diagnosis. Thank you. That's the end of this medical image analysis introduction lecture. For more information, you can check out my website.